Among bacteria, we distinguish between gram-positives and gram-negatives. This classification is based on a staining technique. Today, we show the difference between those two types. Before we start, make sure to subscribe to the channel to not miss out on any new video. Gram-positive and gram-negative bacteria show some differences based on their physical properties. The genome floating around in the cytoplasm is protected by the cell membrane in both types. Around this membrane, a cell wall ensures further protection. The cell wall consists of peptidoglycan. Gram-negatives have a thin layer of peptidoglycan, whereas gram-positives possess a very thick peptidoglycan layer. In addition to the thin cell wall, gram-negatives also have an outer membrane. This is not found on gram-positives. These physical characteristics can be determined under the microscope following the Gram staining technique. Gram staining was published in 1884 and is based on a sequence of chemicals. Starting with using crystal violet, followed by an iodine treatment, washed out with alcohol, and finally counterstained. This procedure leads to gram-positives appearing purple and gram-negatives red or pink. Let's have a more detailed look on how gram staining works based on the cell wall property. At the start, crystal violet molecules pass through the cell wall and stain both types of bacteria purple. The purpose of iodine is to interact with crystal violet ions, and together they form a complex. Iodine is considered the trapping agent. The alcohol is doing two things. The outer membrane of the gram-negative is lost and the crystal violet iodine complexes are washed out as well. The thick peptidoglycan layer of the gram-positive, however, traps the crystal violet. The counterstain, which can be safranin, is taken up by both cells and colors the gram-negatives red or pink. In gram-positives, the dark purple color is stronger and therefore they just appear purple. It is important to determine the physical properties when targeting pathogens with antibiotics. In general, it is much more difficult to treat gram-negatives because of their additional outer membrane. Antibiotic resistance is a serious threat to human civilization. Labs around the whole world are constantly developing new and effective antibiotics. Thanks for watching, make sure to subscribe, and if that video was helpful, leave a like. Bye!